Hi, everyone, and thanks for joining us for episode 748 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I'm looking forward to re-sharing our interview with Dr. David Fitzpatrick, originally released on January 1st of 2018. Listeners, David's research focuses on basic science questions about how the brain works and how it develops. He's a leading expert in systems neuroscience who studies the fundamental organization and development of neural circuits and an area of the brain called the cerebral cortex. This is the largest and most complex area of the brain. And in our conversation, David shares insights and stories from his life and research. So get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and thank you for listening to the People Behind the Science podcast. Listeners, today I am joined by Dr. David Fitzpatrick. So, David, welcome to our show today. How are you?
I'm doing great, Marie. Thank you for inviting me.
Well, thank you for agreeing to join me on this wonderful journey we are going to have today. And I want to take a moment to introduce our listeners to you and tell them a little bit about you before we jump in. So listeners, David is the Chief Executive Officer, Scientific Director, and a research group leader at the Max Planck Florida Institute for Neuroscience. He received his bachelor's degree in biology from Pennsylvania State University and his PhD in Psychology and Neuroscience from Duke University. David then conducted postdoctoral research at the Medical University of South Carolina, and he returned to Duke University afterwards as a member of the faculty. Now, before accepting his current positions at Max Planck Florida Institute, David was the James B. Duke Professor of Neurobiology and Director of the Institute for Brain Sciences at Duke University. He has received numerous awards and honors over the course of his career for his outstanding research and teaching, including the 2011 Ellis Island Medal of Honor from the National Ethnic Coalition of Organizations, the Alfred P. Sloan Research Award, the Cajal Club Cortical Discover Award, the McKnight Neuroscience Investigator Award, and the Excellence in Basic Science Teaching Award from Duke University School of Medicine. And listeners, David is here to chat with us today about his experiences in science and life in general. So David, we want to get to know you both scientifically and personally. And can you start by telling us what you do with all your free time, I'm sure, when you're not doing science?
Free time, what is that?
Right.
So I really enjoy being outside. I really enjoy nature. I love hiking. I love kayaking. I love biking. And something that's actually, I'll call it my hobby, that I really love is photography. So being able to photograph nature, to capture those memories, there's nothing better for me than to be able to look at an image and immediately recall what had happened at that time, etc. It's a wonderful way to reflect on all of the interesting things you've done during your life.
I love it. You've got a little bit of an artistic side to you then.
Yeah, actually I do. I mean, there are people that see my photographs and they get very excited. I think a lot of people understand my photographs better than my neuroscience.
Absolutely. Well, we are going to move on to chat about your neuroscience next, David. So can you tell us how you describe what you do at work to someone who's not familiar with neuroscience?
I start off by telling them how critical the brain is. I'm surprised many times that people don't really think about their brain and the fact that everything that they experience, everything that they're able to think about, their movements, planning, etc., decisions, this is all your brain. And it's funny that people are so preoccupied with the activities of daily living that many times they don't think about that. So first of all, get them on board with the idea that the brain is our everything, and then move beyond that to say we still know relatively little about how this structure works, how this structure develops, and that my goal as a neuroscientist is to dive deep into that question and to provide new insights into how our brains work and how they develop.
Excellent. Well, as a fellow neuro nerd, I'm excited already with this conversation. So you've got my attention.
Okay.
And I know as a scientist, there's a lot of work to get done in a day and it can feel overwhelming sometimes. So I'm always on the lookout for motivational quotes, saying things that can keep me going. So David, do you have a favorite of your own?
Well, yeah. Throughout my scientific career, I've been really focused on basic science. And lots of times, I will say, when you're in a medical school environment and whatnot, as a basic scientist, you feel a little bit like you're out of place, because a lot of the work is translational, it's devoted to specific disorders and that sort of thing. And that's not my shtick. I'm really interested in fundamental understanding of the biology of the brain. And when I got invited to be part of the Max Planck Society, which I will tell you made me incredibly happy. It's quite an honor to be a part of the Max Planck Society. I became aware of a quote from Max Planck that I honestly had never heard of before. And his quote was, "Insight must precede application." Those 4 words really sum up my motivation to be involved in science. And that is, we really need to understand nature at a fundamental level and take that knowledge and use it to address the problems that we have. And for a neuroscientist, the fundamental knowledge we gain can be used to address a host of neurological and psychiatric disorders. But I must say, I'd never seen it stated so eloquently and so simply. Insight must precede application. And I've used that a lot, mainly because I feel like that sums up my view of things.
Absolutely. Well, I think that's an awesome quote. I actually hadn't heard of it either, but I think one of the challenges in being in basic research is trying to communicate to others the importance of it, especially people who are outside of science. And that really does package it up nicely.
It does.
So now I know as a scientist, you go through a lot of different stages of your career, and there's influential people that you meet along the way at each of these stages. Can you look back, and are there any maybe motivational figures or people who really inspired you and kept you going in science?
Yeah, well, actually, there are 2 of them, one early in my career and one later in my career. My graduate advisor, his name was Irving Diamond, and I actually did my graduate work at Duke before going back there on the faculty. And Irving, he was a remarkable person. What got me about him was that in terms of being an experimentalist, I wouldn't say that that was his strength, but he brought a clarity and a criticalness to his thinking that really inspired me as a student. He would teach a course in which he did textual analysis. And I can remember sitting in this course and being a bit scared, right? Because he believed in the Socratic method, which basically means you can get called on at any time to respond to any question. Mm-hmm. And he would take scientific papers apart. He would literally take sentences out of it and ask you, what is the author trying to communicate here? Is what's being said here consistent with the data that's being presented? What more would you like to see beyond what's being stated here, et cetera? And that has stayed with me throughout my career. I think being able to communicate effectively, to say precisely what it is that your data is addressing and how you can interpret it, what the weaknesses are in that interpretation, et cetera. Those are all things that he really turned me on to. Also gave me a hard time about my own writing and forced me to, I think, in many ways, become the critical thinker that has led to the success that I've had as a scientist. The other person, frankly, is Bert Zachman. Bert Zachman is a Nobel laureate. He's also part of the Max Planck Society. And let's just say that he's older than I am, and I'm pretty old. Mm-hmm. And I love getting together with him because he is so engaged in the science. The first thing he says to me when he visits here, he was actually first on the ground here in Florida many years ago, David, what's the most exciting science that's happening in the Institute right now? And then we start talking about it, et cetera. And for me, that's my new role model in a sense. I want to be continuing to be engaged in science at this level as long as I possibly can.
Absolutely. Well, that is awesome. Having these 2 different mentors, focusing one on communication and the other on just kind of keeping that curiosity, that excitement, that enthusiasm alive. I love it.
Exactly.
So where did your excitement and enthusiasm for science begin, David? Can you remember that first memory of getting excited about it?
Yeah. When I went to college, I wasn't sure that I wanted to be a scientist. I was thinking about being a veterinarian. And I got a little bit concerned because I realized as I worked in a veterinary hospital in the summers that it seemed kind of routine. It was kind of symptom, drug, and this and that. And I didn't see the kind of intellectual engagement that I really wanted. And then I had a course at Penn State. I'll never forget. The guy's name was Paul Aplenop. And he taught two courses. One was on animal behavior, and the other was neuroanatomy. And he had gotten his PhD from MIT, and he was an amazing communicator. He made science come alive. And after a course with him, I was like, wow, this is what I want to do. I want to be engaged in study of the brain. I actually did like a rotation in his lab and had hands-on, but it really was his ability to make it come alive, right? Sometimes when you read about science, and I feel bad about the fact that the public many times thinks of science as maybe boring, too difficult to understand. Many times when science is being taught in the schools, it's all about learning how to do math problems and calculus and so on and so forth. Mm-hmm. And lots of people fall off the wagon, if you will, at that point because they don't really understand how useful that is to solve really interesting problems. And Aplenop really got me excited both about science and about the brain. And he deserves a lot of gratitude for getting me going in that direction.
Absolutely. Now, you mentioned the importance of science communication a few times now during our conversation. Can you tell us a little bit about some of the efforts going on at Max Planck to communicate some of the work you're doing there?
We really believe that this is critical to get the word out to the community. And we do this in a variety of different ways. We have a program we call Science Meets Music, where we basically pair up musicians with scientists. I call it a musical sandwich. So we have a musical performance and then about a 20-minute science lecture and then another musical performance. And the public has gotten so excited about these. So we're choosing people who communicate their science in an accessible, exciting way. We will have Nobel laureate Stefan Hell doing one of these in January.
Awesome.
Again, if you can make the science accessible and also convey the excitement that one has in the discovery process, then I think people really get on board. And to some extent, by doing the Science Meets Music, we're trying to get people to understand that art and science are not that far apart. They really aren't. Mm-hmm. That's one of the ways we get people engaged. We have a lot of other programs here. We have programs for high school students in the summer that I'm extremely proud of. We bring students in here. We give them an opportunity to work in a laboratory for 6 weeks. And at the end, they give a presentation on what their project was and what they've learned. And I just sit there and I go, oh my God, I couldn't imagine being in high school, being able to deliver something like that. We also do a lot of work reaching out to the scientific community. We have created something called a Symposium here that we do every other year, where we bring neuroscientists from around the world together for a 2-day conference that focuses on a broad range of fundamental questions about the brain. And that has really become popular with the scientific community. So I think it's important to do both, to be doing what you can to build bridges within the scientific community and building bridges with the public as well.
I absolutely agree. And it sounds like Max Planck is a phenomenal place to work. But can you tell us what drew you there from Duke University?
I thought I would probably spend the rest of my time at Duke. And I was given a wonderful opportunity here in Florida. And if you're a scientist and you're asked to become part of the Max Planck Society, you listen. It's a wonderful organization. There's a motto here, science first. And you noticed that in my title, you mentioned that I'm CEO. Mm-hmm. I smile and almost laugh at that sometimes because as a scientist, I never thought I would be a CEO. In the US, that's the term that's used for my position. In Germany, at one of the Max Planck Institutes there. By the way, there are 83 Max Planck Institutes.
Oh, wow.
Almost all of them are in Europe, most in Germany, in fact. One outside of Europe, and that's here in Florida. So I was being asked to be the founding director of the first institute outside of Europe. That's an amazing challenge, an amazing opportunity. And in the Max Planck Society, when you found an institute, you're responsible for the scientific direction of the institute. You're responsible for recruiting additional directors and research group leaders. So it was an amazing opportunity and one that I simply could not pass up. And we're here in Florida. I came here in 2011. I will be modest and I will say I think we've done very well. The Institute is thriving. Our colleagues in Germany are very pleased with our progress. The science that's coming out of the Institute is world class. It's worked.
Absolutely.
When that happens, it's not one person. I've been fortunate to recruit a number of excellent people, both on the scientific side and administrative side. And it's just all come together in a wonderful way.
Certainly, and I can concur that you are doing phenomenal work there, not only as a leader, but also as a researcher there. So can you tell us a little bit about a project you have going on right now that you are just so excited to be working on?
I am really excited about something that we're calling functional synaptic architecture. So each neuron has a dendritic field where synaptic inputs arrive. Those inputs are integrated. Neurons produce a response that is transmitted to other neurons in a network. And what I find amazing with all the progress that we've made, there isn't one neuron in the brain that we have a complete picture and a complete understanding of that input-output transform. And I think that's key. I think we need to understand, given the variety of neurons there are in the brain, what are the fundamental principles by which neurons transform their inputs into their outputs. I work on visual cortex, and one of the hallmarks of visual cortex, and this is true of other regions of the cortex as well, is that neurons are remarkably selective in their response properties. So neurons in the visual cortex, for example, respond selectively to the presentation of edges of a certain orientation, and edges that move in a certain direction. So a neuron, for example, will respond to a vertical edge moving to the right, but not to that vertical edge moving to the left, and it won't respond to a horizontal edge. And that selectivity is an emergent property of cortical circuits. So what I would like to understand, and what we're busy trying to understand, is how that occurs at a single neuron level. How does a neuron sample all of its inputs and generate these selective response properties? Now, it's not like people haven't been interested in this question for a long time. They have, but we have a phenomenal new set of tools that are really allowing us to dissect the different factors that can contribute to selectivity of neurons. Perhaps your audience is familiar with two-photon in vivo imaging, which allows us to use fluorescent calcium sensors to literally visualize, by changes in fluorescence, changes in calcium that are correlated with changes in the activity of neurons. We've taken this another step, which is to look at single neurons, look at the synapses, the dendritic spines of these neurons, look at their functional properties, and then ask, how are those functional properties, the functional properties of the inputs, related to the output from the neuron? And we have some really interesting observations. The field likes to say, when talking about cortical circuits, like connects with like. It's really easy to remember. Sounds great.
Looks good on a slide. Yeah.
But what's really cool is that we're now able to examine that question at a synaptic level. So we can characterize the functional properties of the inputs to a neuron and compare it with its output. And guess what? Yes, if you look at the inputs to a neuron, you could predict its preferred orientation, and actually, recent work from the lab, its preferred direction of motion. But you find that there are a lot of inputs that are not tuned to the cell's preferred. In fact, you do a very poor job of predicting the tuning of neurons. So when we say neurons prefer, say, vertical, they'll respond to a range of orientations. Some neurons respond to a broad range, some to a narrow range. You can't predict that from the synaptic properties of the synaptic inputs. So that's an important step. It's not just like connects with like. These neurons are doing something else with their input in order to achieve the selectivity that they exhibit. And to put it in a nutshell, one study we've shown is that there's clustering of the synaptic inputs that leads to activation of dendritic nonlinearities that amplify the inputs that are associated with the cell's preferred orientation. So again, this is something that's happening that the neuron is contributing to. Its dendritic field is actually operating on its input to develop its selectivity. And we just discovered, and this is a paper we're working on right now, that when you look at direction selectivity, selectivity for direction of motion, again, you have a lot of inputs that you might say are wrong. In other words, if you look at the cell's output, it responds motion to the right. There are a lot of inputs that actually prefer motion to the left. But what we found is that there's inhibition that comes into the cell that basically reduces response to the null direction. And that inhibition is critical for explaining the selectivity of these cells. Now, again, this is with technology that we've only had recently that allows us to visualize the properties of the inputs and compare it with the outputs, et cetera. I'll stop. You got me going.
I know.
Wait, one last thing. What I'm really excited about now is that we're actually doing experiments where we can not only say what the functional properties of the inputs are, but where they come from. So we're using expansion microscopy, a technique developed by Ed Boyden at MIT. We're labeling inputs. In vivo, we characterize the functional properties of a spine, and then we look to see the source of the inputs to those spines. So we're quite literally describing the input-output function, the properties and the source of the inputs to a particular part of the dendritic field.
This is so phenomenal that you can really just watch these things happening almost in real time and be able to start to understand some of these very fundamental aspects of the nervous system. But I imagine that there's a challenge or two, perhaps, in working in this field, this cutting edge area of neuroscience. And I love chatting about the challenges that researchers face on our program. So, David, do you have a particular challenge you're facing today, or maybe a major failure of the past that you want to share with us today and how you worked through it?
That's a hard one because there are so many challenges. One of the things about being a scientist is that you get to a point where you realize, okay, this isn't working. We got to figure out another way around this and so forth. I mean, I was just talking about spine imaging. We had all sorts of problems initially. Part of the problems we had to do with the technology. So we were using a 2-photon microscope that wasn't designed optimally. So the amount of laser intensity that we had to use to image was a problem for the cells. Basically, they were telling us, we're not so healthy anymore.
Right. All these lasers, yeah.
So we had realized that we had to change the kind of microscope we were using. That made a huge difference. But then when you're talking about the molecular reagents for looking at calcium signals in neurons, I work in non-murine species. Much of the field is devoted to work on mice. I work in two species, the ferret and the tree shrew. Both of these, I believe, offer us really wonderful opportunities to ask questions that we can't ask in the mouse. The mouse is a wonderful model system, and especially with genetic manipulation, it allows you to do amazing things. I'm very jealous of what can be done in the mouse, but there are properties of the cortex. And in fact, I will go on record as having said there are fundamental differences in the organization of the cortex in mouse, in ferrets, in primates, humans, etc. I would love in the long run if we could start understanding what those differences are and how they contribute to the function in those different species. But back to the story, we had to figure out a way to get beautiful expression of these calcium sensors. So we had to start learning how to use viruses. We had to decide on a type of virus. We had to find a suitable promoter. And I'm not telling you about all the time spent trying different things that didn't work. Oh, that doesn't work. Oh, the expression level isn't high enough. Oh, it's so high.
Everything's blotched out. Yeah.
Exactly. The really tricky one was doing sparse. This may sound trivial, but to do dendritic labeling, what you need to do is sparsely label the neurons so that the neuron is sitting there by itself. Mm-hmm. We spent lots of time back and forth, and then we realized that we could take advantage of a molecular technique that others had developed as a Cre-Flex system, inject them at certain concentrations, and by chance, some neurons would be transfected with both the Cre and the Flex, and then they would express the calcium sensor. So when you get to read a paper and you see the beautiful data and everything, we don't talk about these things.
It looks great on paper that everything just worked. Yeah.
Yeah. Just like, oh, yeah, they did this and they did that. And yeah, how did we get there? It was a long, hard road. Dan Wilson, who's a graduate student in my lab, deserves an enormous amount of credit for working through all of the problems and coming up with a technique that actually does what we need.
Certainly. So we don't just want to talk about these tough times, though. I'm glad you shared them with us today. David, do you have a success story, one of these wonderful moments in science you want to share with us today?
Well, since I brought up Dan Wilson, I take great pride in having students and postdocs that come into the lab and then go on to great things. And Dan, he's just a very special person. So he literally just defended his thesis, was it 2 weeks ago now?
Oh, wow.
And when he came in, he was taking a risk. We're a brand new institute. We're just getting the labs up and running. He visited here. We had a good talk. And I could tell that he was really intrigued by the idea of being in a Max Planck environment with access to lots of really cool technologies and whatnot. But he's a graduate student, right? He had never done any of these things. He'd never done 2-photon imaging. He had never done viral injections. I mean, this is all brand new. Mm-hmm. And to see a person come in and go from essentially not knowing all that much about neuroscience to a point where he's leaving here. He's going on to Chris Harvey's lab at Harvard. And I can tell you there were multiple people talking to him, wanting him in their lab.
In high demand, yeah.
And also realizing the impact that he's having on the field. There are multiple people that we are collaborating because Dan has built almost like a network of people that understand what he's able to do and then want to be a part of that. That, to me, is success. And we worry these days about the future of science, right? And it makes me feel wonderful when I see a person like Dan who has the passion, has the intellect, the creativity. He has it all. It makes me feel like when I step aside, there's going to be another generation here that's going to really be carrying the flag of fundamental basic science.
I love it. You're leaving it in good hands. That's awesome.
Yeah.
Well, congratulations to both you and Dan on this exciting success, the recent defense of his thesis. And we've talked about some different aspects of your career, David. And one of the things we haven't mentioned are some of the opportunities you get to travel. And I know you're on the road quite a lot these days. So do you have a favorite place that you travel to for science?
Wow, that's a hard one. I would say that is one of the amazing perks of being a scientist. When I was growing up, I never imagined traveling like I have. I mean, literally, I've been on almost every continent. I got one left. I need to get to Africa one of these days. So what just comes to mind is where I've been most recently. I've been in South America. I've been in Rio. Over the last 2 years, I've been in Brazil. I've been in Argentina, and I've been in Chile. for different meetings. Also, the Max Planck Society partners with some of the institutes and universities there, and I play a role in helping to recruit scientists and mentoring them, etc. And of course, I love visiting places like this. I mean, who wouldn't love visiting these places? Just a wonderful environment, things I've never seen before. Iguazu Falls, I don't know if you've ever heard of that.
I haven't.
I was just blown away by that. And of course, Rio is amazing as well. But what excites me even more than the environment are the young people that I've met in these places that are just so excited about science, so driven, so creative. I mean, you give a talk and you have people standing in line to come up and talk with you and ask you questions about relevant to your talk and everything. And there is a huge number of people, young people in Latin America who are really excited about science. And last month I was in Rio meeting all of these young people who are really excited, but realizing that the economy in Brazil right now is so difficult for science. I mean, there's been huge cutbacks and so on and so forth. So I was really encouraging the potential graduate students and postdocs to be considering going outside of Brazil and considering Max Planck Society, both in Germany, here in Florida, etc. I don't know how to express it, but it was just seeing that energy and feeling the frustration of what's going on there. I think hopefully they will be able to pursue their passion outside of that country. And I'd like to help play a role in that if at all possible.
Absolutely. Well, David, thanks so much for sharing these stories from your own travels. And I love chatting about this human side of science because I think it is an exciting, dynamic, wonderful place to work. And there are a lot of creative and amazing people in science. And when you put us all together, quirky and funny things happen sometimes. So I love sharing some of these stories of fun traditions and just funny memories that people have had. So do you have one that you want to share with us today?
Well, this also just happened to me. I'm going to be in a movie and a documentary that has to do with the development of the brain. And they came through my lab and we had a nice conversation. And I have to admit that I had the conversation and I went on to things, but I knew they were in my lab continuing filming and whatnot. And they just sent me a draft of the movie and I'm looking through it. And I have a postdoc in my lab. I'm very lucky to have this postdoc. His name is Ben Shaw. He's an incredible in vivo patch recordings. He's just phenomenal. So I hear his voice. They're talking with him. And then all of a sudden, the picture changes to Ben's arm. And he rolls up his sleeve. And literally, he has a tattoo of a pyramidal cell on his arm.
Oh, that is awesome.
And I knew about this tattoo. I think I've seen it a couple of times, but I had no idea that was going to find its way into a documentary about brain development.
You're capturing the passion, the enthusiasm, right?
Exactly. This is the kind of people that we're working with that really believe in what they're doing and they're passionate about what they're doing. And who knows, seeing that may encourage some young person to go into neuroscience. And I think it's terrific.
Well, I look forward to watching this documentary, David. I'm going to have to follow up with you to get the details. So now we've talked about some of the big questions that you're answering in your own lab related to the brain. But if you had everything, all of the funding, the staff, the technology, the feasibility, every tool you could dream of, what is the one question you would want to answer first?
The big question for me, I would say, is how does experience shape brain circuits? People are so surprised to realize, even people who have children, that the vast majority of synapses in your cerebral cortex form after birth and at a time when experience can influence that process. Now, we know that there's an increase in the number of synapses. We know that there's a gain and or loss of synapses, and we know that these circuits are built, etc. But we fundamentally still do not understand at multiple scales. And by that, I mean from the level of the network down to the level of the molecules within the synapse, how experience builds the brain, because it does, right? It builds it during development and it continues to build it in maturity. So I feel like that is a big question that we are missing. And I think it's a question that has tremendous relevance for disease for society, for understanding who we are and how we got to be this way. So I know it's kind of big and grand, but for me, that's the big, intriguing question that I would like to contribute a small way to before I pack it in.
Oh, that's awesome. Well, you've chosen a difficult one, David, I must say.
Well, you got to think big, right?
That's right. If there's no challenge, there's no excitement.
Exactly.
So now, I know a lot of people, as they go through their career as scientists, they work with amazing people, and you get great advice from these individuals. And I love to pass this on to our listeners out there. So David, can you tell us what is the best piece of advice you've ever received?
I think really it's advice that multiple people have said this to me throughout my career. And it's really simple. Follow your nose. So much of science is serendipity. And I think maybe people don't appreciate this. We teach what science is in grade school. You have to generate a hypothesis. You have to generate experiments, get results, conclusions, etc. But how do you find what the interesting questions are when we know so little about the brain? So what I like to do is to say to people, yes, have a starting point for a study. Make sure that you are addressing a question and you have the right tools to address that question, but keep your eyes open. You never know what you're going to see. And you will see things that you look at and you go, that's weird. Why did that happen? And honestly, I've had this happen multiple times in my career. Those things that seem odd, didn't fit what your thinking was, etc., end up being really, really exciting, but you have to follow them. You have to have the courage to say, I don't understand this, but I think there's something interesting here, and then really dive deep to pursue it. So follow your nose is the way I sort of sum that up.
That is awesome. Don't ignore the quirks in your data. I like it.
Exactly.
So is there any other last piece of advice or last message you want to leave our listeners with?
Well, I've been asked for advice in the past, and I say to people, listen to your brain, listen to your eyes, and listen to your gut. So when I say listen to your brain, I'm saying basically allow your curiosity to take you down roads that you never imagined you'd be on. And then, as I was just saying, use your eyes, be vigilant and looking for the unexpected. I mean, one way of thinking about this is it's kind of like nature's hints that there's something worth exploring, right? Nature's saying, look over here. And if you're not looking for it, you're going to miss it. And finally, you got to pursue your passion for discovery, even when others don't share your enthusiasm. There are lots of times when people say, that's nothing. Right. Oh, it's just an artifact. But if you are convinced that there might be something more there, stick with it. That's where the discoveries come from. That's where the innovations come from.
Fantastic message to leave our listeners with. Can you tell them where they can go to learn more about you and the amazing work going on at Max Planck?
Sure. I encourage them to visit the Max Planck Florida website, where they can learn all about the scientists here at the Institute. We've also created a Max Planck Neuro website that talks about the neuroscience that's done at a number of the Max Planck Institutes within Germany. In fact, we just had a retreat here in Florida, brought together a number of the neuroscientists from Germany here, and actually also from Latin America, to really build bridges, to really understand what's happening within our society. But if they check out MP Neuro, they'll see the latest discoveries that are coming from the entire Max Planck neuroscience community.
Excellent. Well, listeners, definitely check out those websites. Get caught up on what's going on at Max Planck. And David, thank you so much for joining us on this program to share a piece of your story.
And thank you, Marie. Really appreciate it.
It was a pleasure to chat with you and wonderful to have our listeners here as well today. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.